I'm Tom Hughes, CEO of Navitor Pharmaceuticals. I'd like to tell you a little bit about Navitor. So we're a clinical stage biotechnology company located here in Cambridge, Massachusetts. And we're focused on identifying new therapeutics that target mTORC1 in order to address, among other things, age-related disorders. We have field-leading intellectual property, including a number of different chemical classes that address the target. And we have unique capabilities in mTORC1 biology, in large measure stemming from our relationship with our scientific founder, David Sabatini at the Whitehead Institute here in Cambridge. We have a lead molecule called NV5138 that addresses mTORC1 in the brain and activates mTORC1 in the brain. This is being developed for depression, and that particular program is partnered. We also then have a platform of programs that are targeting the inhibition of mTORC1, and we have the ability to address different organ systems and discrete organ systems owing to the nature of the chemical matter that we're developing. We call our lead molecules navalogs. These are rapalogs that are in a novel class chemically and structurally developed and discovered here at Navitor. So this allows us to approach the mTORC1 node as a platform, if you will, and we're addressing different diseases, particularly those that have relevance to aging biology. So mTORC1, in a nutshell, serves the purpose in all of the cells of our body to integrate signals coming in from nutrients, such as amino acids or glucose, along with growth factors that lead to anabolic processes in the cells. And mTORC1 really provides the permission, if you will, for the cell to carry out these processes that are otherwise expensive in the absence of those specific nutrients, and allows for the balance of anabolic and catabolic processes, or the building and renewal of cellular materials along with their disposal and degradation in a way that matches the needs of the cell and the context in which the cell is operating. There are a number of conditions in which mTORC1 activity becomes dysregulated, and in particular in the setting of aging, as mTORC1 is dysregulated and increases, there's an imbalance of these anabolic and catabolic processes that leads essentially to mischief in these cells, and this often takes the form of that intersection between inflammatory processes and fibrotic processes as well, leading to pathophysiology. Our platform of programs is laid out here. Our lead molecule, as I mentioned, is in the clinic. It's in phase 1, heading toward phase 2, hopefully early next year. Our lead inhibitor, Navalog NV2494, that we'll speak about a little bit more in a moment, addresses the kidney very, very well after oral administration, and so we're developing this for kidney indications. This is currently in IND-enabling activities, and we hope to be in the clinic sometime in the first half, so to speak, or more or less next year. We have additional compounds that are addressing mTORC1 behind that, including additional navalogs. But also we have a range of different targeting concepts that we're pursuing for mTORC1 that should lead to additional chemical matter and additional programs as the company matures. So mTORC1 plays a very central role in a wide range of pathophysiologic processes that lead to aging biology. And we know, for example, that modulation of mTORC1 by inhibiting it or by genetic manipulation in a manner that reduces its activity can impact the evolution and severity of a number of these conditions, at least in experimental models. So whether it's diet or environmental stresses that lead to hyperactivation of mTORC1, this generally drives cellular responses in an adverse manner, leading to loss of stem cell function, leading to cellular senescence, to cells going down to EMT, which leads to myofibroblasts and fibrosis, or a reduction of mitochondrial function. All of these things essentially lead to the dysregulation in the cells of our body as we get older and contribute to the pathology of aging. So if you sort of wind forward, mTORC1 has a very well-established, very well-substantiated role in a range of chronic diseases of aging, including congestive heart failure, type 2 diabetes, chronic kidney disease, Alzheimer's disease, other indications, all of which we believe are very reasonable to address down the road with mTORC1 inhibitors. However, it's very challenging to think about developing a drug that might impact either health span or lifespan, or even from the perspective of a small company such as Navitor, to take on large indications such as diabetic nephropathy or Alzheimer's disease. And so our thought, our strategy, if you will, for developing new agents in this area involves finding what we consider to be gateway indications. These are rare conditions that are very, very clearly mechanistically linked to upregulation of mTORC1, largely through genetic means. So in the case of autosomal dominant polycystic kidney disease, there's a very clear and compelling rationale. Likewise, for congenital epilepsies, where you see hyperactivation of mTORC1 chronically leading to pathology. Here we have the ability to use these very defined, focused indications as gateways to first establish the proof of principle for the compounds, perhaps also to establish a market presence, and then as the programs progress, to expand the label, to expand the use, to expand the benefits that we can drive from our programs as we move forward, whether it's diabetic nephropathy or other cardiometabolic disorders or neurodegenerative disorders such as Alzheimer's. So when we look at NV20494, which is our lead Navilog, this addresses the kidney well, as I mentioned, following oral administration. We're contemplating this for ADPKD, or autosomal dominant polycystic kidney disease. This is a condition of real concern. It leads to shortened lifespan in patients who are affected by it. It's driven in large measure by mutation in a single gene called PKD1. And although there are multiple genetic mutations that occur in PKD1, there are a range of either loss of function or missense mutations that lead to reduction of PC1 expression. This drives cysts in the kidney. This leads to very symptomatic issues, including pain, back pain, fullness, kidney stones, urinary tract, and kidney and liver cyst infections, as well as hypertension, which is a very sort of interesting comorbidity associated with ADPKD. The population is about 4 or so per 10,000. We're about 140,000 affected individuals in the U.S., so it makes it a very attractive indication for a smaller company to work against. And we know that while there are existing therapies that provide some benefit to this patient population, first of all, none of them are disease modifying, nor do they address the underlying pathophysiology that's driven by the PC1 gene mutations or PKD1 gene mutations. And therefore, we believe there's a real interest in developing new agents. So how does mTORC1 patch into ADPKD? What happens is the polycystin gene and another component, a component of mTORC1 called TSC2, sit adjacent on the chromosome. And this, what we know happens is that there's a counter-regulation or a cross-regulation of these two genes in which, where there's a loss of expression of PKD1, this leads to also a loss of function of TSC2, which leads to upregulation of mTORC1 activity. And that mTORC1 activity further leads to a reduction in PC1 expression or PKD1 expression. And so there's a sort of vicious cycle that is carried out in the kidneys of these individuals in which chronic mTORC1 hyperactivation is occurring. And this leads, as I mentioned before, to things like inflammation, fibrosis, but also contributes very mechanistically and directly to the cysts that actually really provide the major pathology in this patient population. So it's been studied already in the sense that rapamycin and everolimus, which are also inhibitors of mTORC1, although they lack the selectivity that our compounds exhibit. And they did show a reduction in cyst volume in humans, as well as in a range of different animal models. But because of the limitation on dosing for these particular compounds related to their non-selectivity for mTORC1 relative to mTORC2, we believe that the actual question wasn't fully addressed or fully tested with these studies, and that there's a real opportunity to evaluate the benefit of a full inhibition of mTORC1 that can be obtained with a compound like ours on the pathology in these patients. And so we believe that there's a real opportunity and a need for selective mTORC1s for the management of autosomal dominant polycystic kidney disease, and we're very interested in advancing our program to test them. So what are the Navalogs? Essentially, they are analogs of rapamycin that take advantage of novel insights that our chemists and our team had identified that gave us the opportunity to develop an entire class of selective inhibitors of mTORC1. We understand how these work. We have studied them in the cell with regard to the function, assembly, and turnover of mTORC1 in the cell. We have crystal structure of the compounds bound to the complex. So we have a very strong, very clear mechanistic understanding of how these molecules work. Like rapalogs, they're very potent inhibitors of mTORC1. They're also inherently more orally active than the existing rapalogs, which is a benefit, we believe. They're more soluble, for example. We see no inhibition of mTORC2, which is the major discriminating function relative to the prior compounds. And this means that we spare the cell division and cytoskeletal functions that really limit the use of the rapalogs because of side effects, whether it's immune suppression, adverse effects on the kidney, or deterioration of metabolic function. Existing rapalogs are really handicapped with regard to their ability to be used in all but the most severe conditions. So we believe that our compounds will exhibit better tolerability. We've seen evidence of that in animals so far. And as I mentioned earlier, we have the ability actually to treat this as a new class of compounds in the sense that we can make many, many analogs. We've made over 150. We've tested them. They're all selective, and they're all covered by fresh composition of matter patent applications that have been filed. So we've tested our lead molecule, NB20494, in a series of models of renal dysfunction. Here's one that we ran a study with a group at Vanderbilt looking at acute kidney injury. This is an ischemic injury that's induced in the kidney. And after the injury abates, essentially the kidney goes on to develop a rather profound fibrosis as well as inflammatory processes. And we were able to show that a month of treatment with 494 at a very well tolerated dose led to a substantial amelioration of the fibrosis and inflammation, which sort of provided for us an initial proof of concept that the compound was working and functional in animals. And what I can say is that everolimus tested head-to-head with this did not show this benefit. We've carried out some initial investigation as well in the setting of autosomal dominant polycystic kidney disease, in such that we've tested the impact of 494 exposure in organoid cultures in which one can generate cysts that are characteristic of the types of cysts that show up in the kidney itself. And if you look across these different wells, you can see that there's a reduction in both the number and size of the cysts in this particular experiment, although they're rather hard to see. It is the case that we can reduce the intensive sort of cyst formation in these particular cells. And we know that it's the tubular epithelial cells of the kidney that are both dysfunctional in the setting of ADPKD, but also that overexpress mTORC1. And this is shown in the lower left, where you see this robust staining of phosphoS6, which is a substrate, downstream substrate of mTORC1, being essentially present in the tubules of these mice. So we have a study that's ongoing now, and it's already beginning to show us some very interesting, very positive findings. We're running a study at the Mayo Clinic looking at the impact of chronic treatment with 494 in the evolution of kidney cysts, and as well as other histological and functional features of the mice. So we're looking to advance the molecule. As I think I said earlier, we're looking to be in the clinic sometime next year. And we're designing a very sort of interesting biomarker-driven validation, clinical validation type of a program in which we'll assess, of course, safety and tolerability of the molecule, but we'll begin to collect information on the impact of the exposure of the drug on mTORC1 activity, as well as potentially on endpoints that are relevant to pathophysiology as well. We have our other additional programs coming behind that. So it's going to be a very exciting couple of years to come for Navitor, and we believe for the mTORC1 field as a whole. And we hope that we have some share of voice, if you will, in the emerging and very interesting narrative around age-related diseases and development of novel therapeutics. So with that, I'll close and just thank you for your attention, and just say, when you're thinking about mTORC1, think about Navitor. We have a lot of very exciting activities going on, and look forward to communicating those with you.